And our first wonderful speaker is Ghosh.
Maybe I'll start before you start your timer. So, good morning, everyone. My name is Ghosh, and I'm one of the research fellows based here at Imperial IX, and I'm funded by the Smith Sciences AI and Science program. I'm a computational neuroscientist by training, but in my work, I use neural network models to think about the brain. Specifically, my work focuses on multisensory integration. In other words, how the brain combines information from our different senses, like vision, hearing, balance, and then uses that information to guide our actions and behavior. When thinking about this process, researchers and neuroscientists mostly picture a network architecture like the one shown on the left up here, in which the information from separate sensory channels, like vision or hearing, passes forward to multisensory neurons, and then on to deeper layers and motor outputs. And in a recent paper, I showed that this architecture is optimal for many of the multisensory tasks which neuroscientists use in the lab to test how animals integrate information from different senses. What that means is that in these tasks, which experimentalists use, you can't perform better than you can with this simple feed-forward architecture. But, somewhat unsurprisingly, when other neuroscientists go and look at the brains of real animals and do detailed anatomical studies, what they found is that the architectures don't look anything like this simple architecture on the left. Instead, like the architecture on the right, they're full of interesting structural details, like skip connections which bypass certain layers, or even feedback connections from later back to earlier layers. To me, this discrepancy between the models we make and the data we observe raises two interesting questions. The first is, when are more complex architectures required? Or phrased differently, like what multisensory tasks require more complex architectures? And the second question is, what makes a better network? So perhaps these architectures would always perform equivalently in terms of accuracy, but maybe different architectures are differently robust to noise, more energy efficient, generalize better, etc. So to explore this, I've developed a series of naturalistic tasks in which neural networks have to perform simple but animal-like behaviors in virtual environments. For example, they have to navigate, forage, and hunt. Once you have tasks like this, though, it's quite difficult to know what sort of network architectures will perform well a priori, or even really to know what sort of architectures to test. So, in my work, I've been using tools from machine learning to automatically generate network architectures which are capable of performing these tasks. Specifically, I've used evolutionary algorithms which work by evaluating and then iterating on or varying network architectures. So in this process, you start with a task you're interested in and a very simple network architecture, and then through an evolutionary process, you discover architectures which are able to perform the task well and might be quite different to the architectures which we would design by hand. One interesting aspect of this approach is that for a given task, you don't just generate one architecture which can solve the task well, you actually end up with a whole space of solutions. So in this context, that means network architectures which are very different, but can all perform the task equally well. So how can we interpret this space of solutions? To do that, I've developed methods for comparing the structure and the function of these evolved solutions. In terms of structure, I use metrics from graph theory to describe the structure of each evolved graph, and then I make comparisons between the networks evolved in different conditions. For example, this graph on the left compares networks evolved in two conditions, which are shown in green and gray, across a range of different graph metrics on the x-axis. And hopefully you can see that some of these metrics are very similar between the two, and some are very different. For example, here the density, which is the fraction of possible connections, is very similar between the two conditions. In terms of function, I then test how well these different networks perform, how well they generalize to new situations, and how robust they are to perturbations. For example, this graph on the right shows fitness, how well the networks perform, as a function of input sensor noise on the x-axis. The black line shows a random motion baseline. If you move randomly in this task, you have low fitness, which is invariant to sensor noise. And then the green and gray lines compare networks with two different architectures. What you can see is that these architectures perform equivalently in low noise settings in which they're evolved, but they diverge as a function of noise, showing in this case that this blue architecture is more robust to noise. So overall, this work is providing insights into the structure of multisensory circuits and more broadly, a general framework for exploring structure-function relationships in neuroscience. Thanks very much for listening.
Our next speaker is Mili Ostojic. There you are.
Hello everyone, I'm Mili, I'm a PhD student here at IX, and I'll be presenting to you this long complicated title which basically just means that I'm going to eavesdrop on birds with drones. So we're focusing on the biodiversity of forests and monitoring those, primarily because there's a lot of different species that live in forests. It's a very important habitat, but also they're under a lot of human pressures like deforestation, agricultural land use, so they're a good case study to investigate. And traditionally, we've used like trapping and tagging to monitor species in forests and other environments, but sort of recently that's been improved upon by the use of audio, ecoacoustic monitoring, which is a taxonomically broad method. Obviously, you can monitor anything that makes noise, so we've got birds, frogs, insects, but we're focusing on birds. And it also can investigate both distributions and dynamics, because we can identify species presence and we can also identify behavior based on the calls that the birds are making. But there are issues with ecoacoustic monitoring that the use of drones can address. So, for example, drones can access remote and inaccessible areas that we can't reach on foot. But also, they can increase the sampling resolution, and this is what we're focusing on. Drones can go to more locations than is feasible for a manual study, and this can improve the sampling resolution. And also, we can reduce the issue of static monitoring, because obviously when you have a sampling design and you just put up all the monitors, they just stay there, regardless of what results you find in the meantime. Whereas with drones, you can move around those locations on the fly, excuse the pun, and we can therefore improve the cost effectiveness and time efficiency. So the main aim is to produce a method for autonomous drone-mounted ecoacoustic monitoring with the aim of improving spatial resolution and also increasing the scalability with live adaptation of the sampling design. And we can use this for biodiversity monitoring, monitoring of specific species, but primarily the one that we're focusing on is monitoring areas with spatial gradients, because we can monitor it on a much finer scale with the increased spatial resolution and determine exactly where a species might change its distribution depending on the environmental gradient. So our approach is to have a drone that flies into a forest, lands at a predetermined location, records while landed, and then moves on to the next location, and so on. So we'll be recording for shorter periods of time at more locations than you would in a manual study, because with a manual study you have a static low power sensor, you can leave it for weeks or even months, but the sampling design is fixed. Whereas with this we have many more locations but with less time at each, so we'll be investigating whether that trade-off is worth it. And another advantage is that we don't have self-noise in the recordings. Drones are very, very loud, so we're only recording when the drone is landed with its motor switched off, so we eliminate that problem. And these are the results of a preliminary investigation we did with the manually operated drone. So as you can see, there is a spatial gradient of detection that emerges. This was done in Silwood Park, which is one of the Imperial College campuses. And we had the drone fly into 3 different forested locations at 2 places each. And then we also had a static sensor which provided the 1.5 hours and then also simulated a third drone location for half an hour recording. So we had 3 locations of inside the forest, near the forest edge, and at the forest edge. You can see in Merton's Acres especially, you can see that spatial gradient. And you can also kind of see it in Elm Slope, but there's less of a significant difference between near the forest edge and the forest edge, and that's because we had difficulty flying the drone into the dense area, so they're both pretty much at the outskirts. So one thing that will improve in future studies is to use an autonomous drone that won't face the same issues that we had when manually flying it. And then in Gunnis's thicket, you can see that there is sort of a spatial gradient, but there is also a bit of an outlier there, and that's probably because of the limited sampling size and duration. So there might have been like a very short temporal discrepancy, like a predator going past and all the birds raising an alarm call, that would be smoothed over if we recorded for longer periods of time. So obviously when we do larger scale studies, we'll be recording for much longer than half an hour at each place, but this is sort of a proof of concept. So we have shown that when investigating spatial gradients, we do, the advantage of increasing the spatial resolution does outweigh the reduction in temporal resolution, and we do have a proof of concept system that we can use and improve on and increase the features over time. Thank you for listening.
Benedikt Maier.
Hi. Can you switch to, I don't know. Oh, I just press forward. Okay. Yeah, hi, thanks. I'm Benedikt. I'm talking about how we can disentangle the collisions at the Large Hadron Collider. So what I'm doing at the Large Hadron Collider, we're trying to answer the big open questions in physics, right? What's the nature of dark matter that we know from astrophysical observations? Where does the apparent matter-antimatter symmetry come from in the universe? We have way more matter than this antimatter symmetry. Antimatter wouldn't have to be this way. Where does this insane level of fine-tuning come from in our particles? The Higgs boson is much lighter than it would have to be according to first principles. So at the LHC, we're colliding bunches of protons, each bunch containing 100 billion of the protons every 25 nanoseconds. We're hoping to produce new exotic particles that would answer these questions, and we're recording these particles, hopefully, with the CMS detector. So as we collide the bunches of protons, it's not that only one proton interacts with one proton from the other bunch at a time, but in fact we get on average about 80 simultaneous proton-proton collisions. At the high luminosity phase of the LHC in 5 years, this will go up to 140, even to the extremes of 200 sometimes. And typically there's one collision among those that will be more interesting, that will be the most energetic, looks the most interesting to us, that we want to analyze deeply and closely. And the rest is just junk and what we call pileup, and so basically clouding the picture of the main interaction. So the challenge really becomes to disentangle the products of these proton collisions, what we dub or what we call and refer to as pileup mitigation. So let's look at what we have and what we want. Certainly we have the sequence or a list of particles that we record with our detector, characterized by momentum of the particle that we measure, by angles in the detector, how they're orientated. Now, what we want to disentangle them is basically identify context in this list of particles, context in the sequence of particles. For instance, identify the particles that come from the main primary interaction that we're most interested in, and identify the junk pileup particles accordingly. So if we think, what can we use to identify context? Well, certainly since the advent of ChatGPT, we know that transformers are very well suited to accomplish this task, because they really, at the core of transformers, is a concept called attention that really allows to let the members of the sequence talk to each other, hear each other out, see how much they have in common, and thereby identify a common origin. And in our case, that's ideally suited to identify the particles that really come from the same, namely from the main interaction. So I've developed this attention-based mechanisms over the last 2 years. They were featured on popular physics blogs, also on Twitter. Got a response that to this day I don't know what it means, really. By the way, the sequence sometimes is so long that we cannot do full attention, let each particle talk to each other particle, because it would be computationally not feasible, but we have to play with sparse transformers. But what's nice is that this IX research fellowship allowed me to put this into action and apply it to actual data recorded with the CMS detector, and I will show you the results, preliminary results here. What you see in the left plot is as a function of how many pileup collisions, how many simultaneous collisions we have in the bunch crossings. Basically, the title probably got swallowed in the export to PDF. Anyway, it's the measurement resolution with which basically how precisely and accurately I can disentangle the particles. I put the arrow there, smaller is better, And as you see, as I go through the years of the state of the art that CMS, our detector, our collaboration, employed to solve the problem of pilot mitigation, over the years you got better and better, and my attention-based algorithms are beating the current state of the art that we're currently employing at the LEC Run 3 that's currently ongoing. And also what's nice in the if you compare simulation to data that you're often doing to make statements on whether there's new particles or not, you don't see any artifacts introduced by the algorithm that would be caused by, misassigning particles and would suddenly imbalance your events when you put them together and reassemble them. So, concluding. The key concepts from natural language processing are very useful for us. I showed you one example. We can really use them to increase the sensitivity for searches for new physics. And yeah, there will be a public result soon that's made possible through this IX fellowship. So thank you very much.
Thank you. So it's Alice Malivert.
Hi everyone, I'm Alice. I'm a Schmidt postdoctoral fellow here too, and I'm working on plant biology. So I'm trying to build a tool to detect drought stress from plant pictures. So drought stress is one of the most important reasons for crop losses worldwide, and particularly in low and middle income countries, where it's responsible for the highest percentage of crop losses. And so it's important to be able to detect drought stress effects in plants, to be able to find management techniques and some different crop varieties, some breeds, that can resist better drought stress. So now, how can we quantify drought stress in an efficient manner, especially if you want to apply it to low and middle income countries? So we have different range of techniques. For example, there are field techniques, which are fast and cost efficient and low effort because you just look at the crops and see which one look like they are drying. But as you may know, if you have plants at home, sometimes it's quite subjective whether a plant is drying or not. And it's also very qualitative and it requires training. So we also have lab-based techniques. For example, you can look at gene expression, if drought response genes are expressed in the plant or not. And these methods are usually very objective and they're very quantitative, but they're never fast, never cost efficient, and they require a lot of effort and training. So now we are trying to get the best of both worlds, and to find a method that would be fast, cost efficient, low effort, no training, objective, and quantitative. And so for that, we take a leaf out of the farmer's book and we just look at plants. And so I'm trying to make an algorithm that will be able to detect a drought stress from plant pictures. And I want to use just normal RGB pictures taken from a cell phone, so that kind of quality, because that's the kind of equipment that most farmers will have, even in low and middle income countries. And so I'm taking pictures and I'm trying to place them on a drought stress scale. So my method, first I had to create a data set, because actually there's no data set with both the pictures and the physiological measurements that I was looking for. So these physiological measurements are various lab measurements of how drought stress is impacting the plant. And so I took this data set with the pictures, so by drying plants and over time taking pictures and the physiological measurements. And since it's quite a small data set, because I'm taking it myself, I have to go through a run of data augmentation, and I'm using transfer learning with quite classical convolutional neural networks that can classify images. I have to go through a step of labeling, which I will go back to in a minute, which is non-trivial. And then once I have my classification algorithm, I would like to get an explainable model, for example, with saliency maps, to see what part of the picture was used to detect drought stress. And then ideally I would like to have a generalization of my model because I'm working on a small plant called Arabidopsis thaliana that's used a lot in biology and that's basically useless. But it grows fast. And it's in the family of the cabbage and the mustard. So similar kind of methods with plant diseases have shown that it was highly transferable to other species. So I hope that my model will also be transferable to species with similar leaf shapes in the same family. And so the labeling, which is non-trivial because actually drought stress is not something you can measure. There's not like one instrument that will give you a drought stress on a scale. But I can measure physiological parameters, which are how the plant reacts to drought. And so I have a lot of them, and how can I use them as a scale of drought stress? First, I'm selecting the most important parameters, the most important features, and for that I'm using an algorithm called MRMR, which selects the most important features while keeping a low redundancy. Because I have a lot of redundant parameters. For example, all the photosynthetic parameters are quite redundant. And then from these selected features I use them to apply non-supervised machine learning algorithm clustering so that I can see in the space of these features which data points are closer together and establish groups of data that look alike. And so I'm using these clusters as labels for my labeling, for my classification in the end. So if you want to know more about it, you can come and see my poster over there. Thank you.
And Emmanuel Akinrintoyo is the next speaker. There you go.
Good morning, everyone. I'm Emmanuel, a PhD here at IX. I'll be talking about the use of robotics for providing cognitive stimulation therapy for people with dementia. So dementia today is a global healthcare challenge. There's over 55 million people globally with dementia, and there's no cure for it. And so one of the key symptoms of dementia is that it causes cognitive degeneration, basically. So a lot of people with dementia would lose their cognition over time. And this basically leads to what we call reality disorientation. And reality disorientation means they lose synchronization with time, with the person who they are, and they also lose synchronization with their environment in general. So a lot of researchers have found that non-pharmacological interventions are the most effective for alleviating some of the symptoms of dementia. And that's what we're looking at in this research. ICST, which is individual cognitive stimulation therapy, is one of the foremost evidence-based intervention for dementia, and it was designed to be delivered by family carers who would stay in home and deliver this kind of activities. So I've shown one there on the screen, categorization of objects, comparing objects, which ones are similar, what do the images represent, and so on. And they would have these activities in home on a twice a week for 45 minutes. But the problem is that a lot of those caregivers do not have the time to deliver this kind of activities because they have to cater for their loved one on a regular. So there's that caregiving responsibility they have to cater for. And people with dementia also do not like to do this with their family members because there's that element of what happens if I miss it. So they feel stigmatized in that regard. So research has shown that technology is a potential solution for this. In our research, we're looking at a 3-in-1 robotic solution that would provide be of long-term use, that would provide ICST for 30 minutes twice daily. So they get to do this. They have the system in the home 24/7, and they're able to do this twice whenever they want to. And it would also be able to detect disorientation, meaning it would know when they lose synchronization with the time of day or their environment. And we're also looking at an NLP system that would be able to understand dementia speech. So dementia speech is very different from that of a healthy person. So there's a lot of nuances such as repetitions, pauses, and so on in dementia speech. But a lot of the current systems do not cater for this today. So we're looking at incorporating that into our system. So our system basically comprises of a MISTI robot, which is the white shining device there on the right, and it has a tablet that shows images, text to aid the interaction. We have a mini computer there that also serves as a local server to provide in-home data storage and processing. So Misty is able to like engage in an interaction with the user, understand what the user is saying, provide feedback, and engage them cognitively to provide cognitive stimulation. So we're doing this with a number of independent living facilities across the UK. So we're partnering them by consulting with them to get feedback on design ideas, requirements, and so on. So we intend to deploy this in-home very soon. Currently, we're doing a one-day study where we go into the care home and then we put the robot in front of the people basically to like see how they get on with the system and then collect some feedback from them. So to end, a number of activities we have currently include popular faces. So it's able to show you a lot of celebrities and pictures of popular people globally and engage you in a discussion on them to like provide stimulation. We have proverbs as well, or popular places and so on. So currently we envision having this in-home in the next few months to provide stimulation for people with dementia. So thank you for listening this morning. Yeah.
Our next speaker is Elli Heyes.
Okay, hello. My name's Ellie. I'm one of the newest fellows at IX. And just in one sentence, what my research is using the tools of machine learning to study the geometry of space-time in string theory. So in 2024, we have a state of affairs in physics. So we have two models that work very well in their respective domains, but don't work well when you try and fit them together. So on the one hand, we have the standard model of particle physics. This is a quantum theory describing all of the elementary particles and the three non-gravitational forces. And it's a very, very good theory in that it matches experimental data to a very high degree of accuracy, as Benedikt was alluding to earlier. But it's not a complete theory of physics because it doesn't describe gravity. And then on the other hand, we have general relativity, which is Einstein's theory, which describes gravity as a geometric property of space-time. And yeah, as I said, these two models, usually you only really need one, but in cases like the center of a black hole or the beginning of the Big Bang, you need both theories together, and when you try and put them together, you get all sorts of ugly things happening. So ideally, we would like a complete theory of physics, and that's what we refer to as a theory of everything. And currently, at least, string theory is the most promising candidate we have for a theory of everything. So the main premise of the theory is that rather than everything being made up of lots of zero-dimensional point particles, instead, everything is made up of the single one-dimensional string. And then you get the different particle properties that we observe in experiments, just as different vibrational modes of this string. And one of the big caveats that string theory comes with is that it's a theory that only makes sense if you have extra spatial dimensions. So essentially what my research is studying the geometry of these extra spatial dimensions and seeing what kind of physics you get out of it. So specifically in superstring theory, we have 10 dimensions of space-time, so 1 dimension of time and 9 dimensions of space. But we're used to 1 dimension of time and 3 dimensions of space, so we say that these extra spatial dimensions exist on some small, compact geometry, and they're so small that we just don't notice them. So in general, the geometry of this internal space influences the kind of physics you get, so the kind of masses of particles or the strength of forces, etc. And yeah, so we apply some conditions on this internal space from physics, and this imposes that this internal geometry is what mathematicians call a Clavier manifold. So why do we want to use AI to study these things? So one of the problems that we have is, as I said, this internal geometry must be a Clavier manifold, but we have lots of different possibilities for what Clavier manifold it can be. So one way we're using AI is to identify the good ones. So by good, I mean ones that would give rise to physics that matches what we observe in experiments. The next problem that we have is that we need this equation on this Calabi-Yau manifold, which we call a Ricci flat metric. And this essentially defines a notion of distance on the manifold. And we need that in order to compute things like the masses of particles. But there's no analytical way of getting this metric. So again, this is an area where we're using machine learning to approximate this metric. And then even if we have the manifold and we have the metric, sometimes in certain string models, it's still then very difficult to actually compute the physics. So here's another area where we're inputting tools from AI. So just a couple of specific examples of my research. One thing I've been doing is using evolutionary algorithms, so genetic algorithms, to generate these Calabi-Yau manifolds with the specific geometric properties that give us the right physics properties that we want. Another one, as I said, is approximating these Ricci flat metrics on a Calabi-Yau manifold using neural networks. And finally, as I said a couple of slides ago, we impose some conditions from physics onto our manifold. One of the conditions that we often like to impose is something called supersymmetry. And so one thing I'm now studying, having joined Imperial, is what kind of geometric spaces we can have if we impose the right amount of supersymmetry. And that's it. Thank you very much.
Amazing. So Sophia Yaliraki, yeah, you have.
Thank you. This one? Okay. Okay, so I'm in chemistry and IX. In my group, we like to import old and new math to think of chemistry in a different way. In particular, what interests me is how to abstract a chemical fingerprint at different scales, and in particular in biological problems. You don't need the absolute detail in complete accuracy of the 10th significant figure to understand this, but neither you can abstract the molecule as a sphere and see the differences, the very nuanced differences. So I'll focus on one snapshot of how we try and solve this problem through constructing atomistic graphs that allow us to get insight at different scales extremely efficiently with minimal data in a simple computer, and at the same time try to quantify and be quite rigorous what we understand and what we don't understand. Why graphs? Well, graphs have been introduced for a very long time, a very elegant mathematical objects with a lot of rich math that we can use. But the way we actually use them is quite a bit different to how most people do, in that we're really insistent on putting in the physical-chemical detail. So we want to put together as much of the geometry and topology of the molecule as well as its energetics in the way to do that. And the way after we have those graphs that contain this information, we can do a lot of different processes on them. We have been focusing on using diffusions and nonlinear approaches on them to extract information at different scales quite efficiently. So just briefly to explain why this is a little bit different, we start with some snapshot information of the molecule. It can be as small as big as you like. It doesn't need to be very accurate. And then we try to both obtain the edges and the edges for these interactions, for both strong and weak interactions. For those of you who understand chemistry, we have been able to capture the hydrophobic effect at all scales. Usually you need different levels of theory for the small scale and the large scale, but because we have the geometry with some geometrical dimensionality reduction technique, we're able to capture that. And then we also weigh those edges. The energetics come in. It's very important. If you don't weigh them, we can't get a lot of the information we can. Also, we remain efficient. This grows linearly with the size of the system, so we can do the biggest biological systems, for example, without any problem. And then we have a series. You can ask lots of different questions depending on what you are interested in, from how did this molecule work at different scales. With multi-scale community detection, we can actually compare with many different experimental techniques with one calculation, which is quite nice. Or we can look at how information signaling goes through this molecule at far away ways with the different techniques. Most importantly, we really try to quantify the uncertainty, how good these results are away from randomness and so on, so that we can have some validity on what we are finding, or if we don't find anything. I'm a big believer in saying, you know, this method is not good here, I can't find anything. So you're not always going to find something interesting. I think this is as important as finding something new. These methods are quite important in finding for problems that have multiple pathways that are nonlinear, and for processes that appear very far away from the local chemistry and physics that we are interested in. They are good for anisotropic systems, where a lot of biology is based on. I'll just give an example on a problem of allosteric and allosteric drug design. Nature uses allostery, which is communicating very far away from where some chemistry is happening. We don't understand how that happens, and we don't know how to use it, and we like to use it for drug discovery, because most drugs, almost all drugs, are in what is called the target and not far away. So these approaches actually lead to very good results. We have benchmarked, and at the moment we have a state-of-the-art method for this, and that has led to actually new drugs being found and used in the clinic. We can also optimize those paths to, again, not only find new targets for allostery, but also design different kinds of molecules that we need desperately to get away from this problem of resistance. You know, penicillin was invented at Imperial 100 years ago. It's time to get to the new space, both in targets and molecules. We hope Imperial will be there too. Thank you.